Thank you for coming to my virtual poster, Can Corniness Be Measured? A Tale of Two Frequencies, where we studied corn stover with 2 MHz and 250 MHz NMR and determined that both can measure water absorption in corn stover, but they provide slightly different information. This study opens the door to portable NMR, where the 2 MHz field strengths used here rival the lowest ever for agricultural wastes. They are also on par with portable well logging systems, therefore indicating that there's clear potential to measure wastes in the field with NMR. An example of this is shown here, where you could stick a portable system into a bale of agricultural waste to determine moisture content and maybe even characterize your material. Another example would be to integrate it into a process and take quality control measurements. For example, you could use it to monitor enzymatic hydrolysis progress. Next, let's zoom out and talk about why this matters. Fossil feedstocks are commonly used to produce fuels and goods, but they result in greenhouse gas emissions. Food crops like corn provide a renewable alternative with reduced greenhouse gas emissions, but using them for this purpose increases the cost of food. Yet another alternative is to break down agricultural wastes into sugar building blocks for use as precursors for fuels and goods. The problem with this is that it's not really very economically feasible yet. So we're using NMR relaxometry to better understand these systems to hopefully reduce the costs of breakdown. Particularly, we're using T1 and T2 relaxation. And we're taking advantage of the fact that they have slightly different frequency and correlation time dependencies. We're utilizing these slight differences to determine complex molecular information about these systems. Our measurement is a T1, T2 correlation sequence, and it results in a plot that looks like this, where on the x-axis you have T2, and on the y you have T1. There's a dashed line showing when T1 equals T2, known as the parity line. Things close to the parity line are amorphous solids and liquids, and they display rapid molecular dynamics. Things far away from this line are crystalline or ordered solids or viscous liquids, and they display much more restricted and slower molecular dynamics. Looking at our data, on the left we have 250 megahertz data, where you can see that with hydration, the major population shifts towards the parity line. On the right, we have 2 megahertz data. This data, with hydration, shifts towards the line, but mostly up and along it. These differences in these trends indicate that we're getting slightly different information about our system with these two field strengths. Another way to view this data is to integrate it along the diagonals to produce a ratio of T1 over T2, and we can see how this ratio changes with hydration. Again, we see a shift towards the parity line, or a T1 over T2 value of 1, and we see this with both field strengths. Another way to look at this data is to plot an average T1 over T2 ratio with moisture content, where we see that at 250 megahertz, this ratio continually decreases with hydration. But at 2 megahertz, this data follows a U-shaped trend, decreasing at first before plateauing, and then eventually even increasing a little bit with continued hydration. This again demonstrates that the two field strengths provide slightly different information. A final way to look at this is to plot an average T2 value with moisture content, where we see that the two field strengths line up pretty nicely. So to conclude, we've demonstrated that 2 megahertz and 250 megahertz NMR can measure water absorption in corn stover, but they provide slightly different information. And this is important to recognize if you're going to make any of these measurements yourself. You need to be sure of what you're measuring, and so this requires knowledge of the two motional regimes with NMR. So this work also lays the groundwork for some of our future works, where this is kind of a preliminary data set, but what we've done is we've monitored enzymatic hydrolysis time on cob. So we have both raw and then 4 pretreatment conditions. And you'll notice with this is that the mild pretreatments don't look very different from the raw material, but the severe 10% alkaline and liquid hot water 190 pretreatments show quite a bit of difference. And in particular, this small peak at short T2 relaxation time seems to disappear before 24 hours. We believe that this may be correlated to enzymatic hydrolysis progress, but need to do further work to verify this first. With that, that's essentially the end of my presentation. So if you have any questions, please write them down and leave your contact information, or you can contact me at any of these links shown here. Thank you.